So now we come to the main one, which Anu is going to talk about, which he presented as a late-breaking trial of the transcriptomic signature and predictors of evolutive curve added to maximum statin therapy based on the intracoronary plaque characteristic yellow 3 study. Anu?
Yeah, this was presented in AHA as a late-breaking clinical science. So if you, oh, study was actually sponsored by Amgen and partially by Abbott Vascular. So the thing, despite high intensity statin therapy, a considerable residual risk of cardiovascular event remains in patients with CAD. And PCSK9 inhibitor has reduced this residual risk. The aim of Yellow 3 was to assess the effect of 26 weeks of evolocumab on coronary plaque morphology using intravascular imaging and peripheral blood mononuclear cell gene expression analysis in patients with the stable CAD. So we took patients with stable CAD who were on maximum tolerated statin therapy. They underwent angiogram. Non-obstructive disease was identified. They then underwent OCT to understand lipid-rich plaque, who also had minimal fibrous cap of less than 120 microns. So lipid-rich plaque means the lipid arc had to be more than 90 degrees. They were enrolled in the study. After that, they just had a NIRS IVAS done. All of the patients got evolocumab 140 milligram every 2 weeks in addition to their statin therapy for a total of 26 weeks. 110 patients were enrolled. When they came back for a follow-up after 26 weeks, we repeated OCT NIRS IVAS of the study lesion. We had blood collected at baseline and follow-up for PBMC analysis. So the primary endpoint was change in fibrous cap thickness and change in the maximum LCBI of 4 millimeter, and that one I presented in ACC of 2023. Then the changes in PBMC gene expression is one I presented at the AHA that just happened. So the total cholesterol and LDL between baseline to follow-up, if you see there, significant decrease in cholesterol and LDL. But between baseline to follow-up, there was no change in C-reactive protein. So the primary imaging endpoint as shown here, if you see the fibrous cap thickness, the entire group or the entire cohort, a significant improvement in the fibrous cap when they came back for follow-up. Similarly, a significant decrease in maximum LCBI of 4 millimeters of the NIRS. So baseline 782 of the lipid LCBI came down to 235. We call it a melting butter sign. We then identified who are the patients who are responders. So responders were patients with increased fibrous cap thickness and a decrease in maximum LCBI at follow-up. So there were 80% responders and in fibrous cap and 76% in LCBI. Non-responders were identified as patients who had no change in their fibrous cap when they came back for follow-up, or no change in their LCBI, or most people had both of it. So, but if you see, as shown in this example, fibrous cap is the same 50 micron, LCBI actually increased a little bit from 586 to 690, but look at their LDL. LDL was 78 at baseline. On therapy, they became 7. So you take a biochemical change, you see that LDL has changed. So this is what we think, that in these patients, that there is, okay, we are treating the patient, LDL has changed and they will not have any further event. But you can see about 20% of them non-responders when they come, when we see their plaque inside the body. The question is, was there a change in the total cholesterol and LDL between follow-up and baseline and follow-up in the responders versus non-responders as shown here? No, everyone responded biochemically, but there is something different in their plaque. So what is it that is different in these patients? A total of 104 patients had imaging and transcriptomic data available at both baseline and follow-up. what we did was the bulk RNA sequencing, okay, not individual cell, but the complete bulk sequencing of the PBMC. 1493 differentially expressed genes were identified. And as you see there in the red, 922 were upregulated and 571 were downregulated. And with the significant p-value, you can see at the side the significant p-value between baseline to follow-up, and you see the heat map. The heat map is the one that shows that between baseline to follow-up about the various genes. Also, what we had done was the ingenuity pathway analysis to understand the key molecular pathway associated with plaque stabilization after 26 weeks of evolocumab. And we use the canonical pathway to of the 1492 genes. And what we found, the most important genes that were upregulated, as you see there, was the orange bar. And those were the genes that were associated with pathway responsible for electron transport, ATP synthesis, oxidative phosphorylation, mitochondrial translation, and crystal formation. All of these upregulated genes essentially showed that there was an improvement in mitochondrial function. And the so-called mitochondrial dysfunction pathway, if you see that in the blue, it was downregulated. So the other downregulated pathways were binding and uptake of ligand by scavenger receptor phagocytosis, cell surface interaction at the vascular wall, which is the most important pathway responsible for the plaque formation. And I will go over it a little later. And then lot of immune cell interaction between various, I would say, immune cells, all responsible for inflammation. So these were all these blue pathways. These pathways were downregulated. The other important upregulated pathway was the pathway responsible for protein synthesis and cellular response to stress, as shown by the eukaryotic translocation initiation. This was responsible for cell survival. When cell survival improves, apoptosis decreases. When there is a decrease in apoptosis, there is decreased plaque formation. So other gene associated with increase in fibrous cap in the entire group, what we found was 15 upregulated genes, and most of these upregulated genes, as you see that in that what we call as a co-expression network analysis, gene responsible for smooth muscle cell contraction, which is very important for plaque stabilization. Other downregulated pathways were pathways responsible for IL-6 and TGF-beta signaling. Both these pathways are responsible for inflammation. So so-called inflammation responsible pathway was downregulated. Now the question comes, what is the difference between fibrous cap responders versus non-responders? So we analyzed them separately. So fibrous cap responders had 2068 totally differentially expressed gene. Of that, 90 significant pathway. All are orange pathway with significant p-values. And what were those pathway? Like we showed, is increased mitochondrial function, cellular response to stress, and increased protein synthesis. All this is important for the cell survival and decreased plaque. What was the issue with the non-responders? 301 genes were identified with 20 significant pathways. And of that 20 significant pathway, what we found was that X should be on the mitochondrial function, that the mitochondrial function was no different in non-responders. But the neutrophil degranulation pathway is upregulated. You see that the orange bar on the top, that equal to inflammation. And the PCV inflammatory pathway was downregulated. That pathway is responsible to decrease the inflammation. Then we decided, why is it that the responders have a better fibrous cap response? So we did a machine learning model to predict why the patients had a better FC fibrous cap response. So there were 38 baseline genes that we identified under the ROC of 0.97. And these genes were mostly the genes associated with inflammation, wound healing, and mitochondrial function. Similarly, the genes that were responsible for LCBI reduction in this machine learning model, the prediction on the ROC was 0.85, and 71 baseline genes were identified responsible for inflammation, wound healing pathway, mitochondrial function, and extracellular matrix organization. So the common pathway between decrease in LCBI and improvement in fibrous cap in these patients was interleukin 1 receptor type 2, which was associated with M2 macrophage. And we all know M2 macrophage is a better macrophage compared to M1, which is responsible for inflammation. MMP9, that was another gene, and a CA4. And all of this essentially means the inflammation, neutrophil function, and mitochondrial function. That inflammation decreased, improvement in neutrophil function, and improvement in mitochondrial function. That's what we saw. So this is animation essentially depicting the plaque regression. So if you see here, LDL comes in the serum. So then there is monocyte. Okay, the monocyte comes and adheres to the vessel wall. The monocyte undergoes phagocytosis, and then lipid uptake happens. Then there is foam cell formation. When all these things happen, increase inflammation and M1. What happens inside the mitochondria? Inside the mitochondria, the reactive oxygen species ROS increases. Because of that, the mitochondrial function decreases with decreased protein synthesis, and you see a downstream effect. All that happens. So we also know when all this happens, apoptosis goes up. When this happens, the lipid continues to build, the smooth muscle cell contractility decreases, fibrous cap becomes thin. You give evolocumab. First thing that changes is the monocyte addition stops, the lipid uptake, foam cell, everything goes down, inflammation changes, becomes M2 macrophage. Now mitochondrial function improves, and also protein synthesis increases. With all these changes that happens, means everything is reversed. Apoptosis goes down, and what happens is the extracellular matrix degradation goes down, smooth muscle cell contractility improves, and the fibrous caps become thick, and the so-called lipid starts going down. So whatever genetic changes that we saw in their gene expression transcriptomic changes, we saw that in the phenotypic changes, which was the imaging, that essentially the lipid-rich core decreased and the fibrous cap increased and the macrophages decreased. So this one was the first of its kind study. So the conclusion of yellow 3 was, in patients with stable CAD on maximally tolerated statin therapy, more intensive lipid lowering with addition of evolocumab resulted in beneficial changes in lesion morphology. For the first time, this clinical study demonstrated an association of plaque stabilization on imaging and improved PBMC mitochondrial function. So reduced monocyte adhesion and transmigration, decreased lipid uptake, recovery of smooth muscle cell contractility, decreased apoptosis contributed to plaque stabilization. So machine learning model predicted individual fibrous cap and LCBI response to evolocumab using baseline gene signature with very high accuracy. The models could help identify patients who might require alternative therapy and have development of personalized approach for treating CAD. This was the entire yellow team from Mount Sinai Cath Lab.